Assalamu alaikum everyone. Myself Salma Farooq. I'm a scholar, microbiology from Guwahati University of Science and Technology. Today I present the topic of synthesis of zinc oxide nanoparticle using zinc officially legume and their antibacterial activity against multi-resistant bacteria. Now these are the table content like introduction, rationale, objective, material method, result, and conclusion. We discuss all of these steps. First of all, we discuss introduction. In introduction, antibiotic resistance by microorganisms is a global issue that needs proper attention to resolve, as most of the deaths occur due to the infection caused by MDR bacteria. These pathogenic organisms like Escherichia coli, Staphylococcus, Pseudomonas aeruginosa, Klebsiella, and Shigella are responsible for bacterial infection in human beings as well as in animals. So nanotechnology is one of the key areas in modern research. Among the metal oxide nanoparticles, zinc oxide is interesting because it's exhibiting high catalytic efficiency and are used frequently as antimicrobial agents. So the green synthesis using plant extracts are commonly preferred for the synthesis of zinc oxide nanoparticles due to the environmental friendly nature, cost effectiveness, and safe nature for the human therapeutic use. So this is a British new. The present research study focuses on synthesis of zinc oxide nanoparticles from the legume extract of Zingiber artificial and then determine its potential to enhance the activity of commercially available antibiotic against MDR bacterial isolates. Also, these are objectives. Number one, fabricate and characterize zinc oxide nanoparticles using Zingiber artificial legumes. Number two, determine the antibacterial activity of zinc oxide nanoparticle against MDR bacterial isolate. Number three, avoid of nanoparticles that used antibiotic for enhancing antibacterial activity. Now, the next one is material method, collection of multi-drug resistant bacterial isolates taken from the culture collection of the Department of Microbiology Research Laboratory. Second one is collection and processing of zinc official legume. So the ginger was cut with the help of knife and dried, sheeted dry for 1 week. So the dry legume blended into powder form. So the extracted powder kept in it and water for phytochemical analysis. So the qualitative analysis of Zingiber officinal legume like anthraxine, equus, and methanol were analyzed for different phytochemicals constitute like alkaloid, flavonoid, and polyphenolic compound using standard procedure. So the fourth one is preparation of zinc oxide nanoparticle because zinc acetate and dehydrate 0.1 mole were taken and dissolved in 50 ml water, then dissolve 1 gram of extract. So for maintenance of pH, then add few drop of anions and place in a shaker for 2 hours. For after shaking, place in centrifuge and then placed in incubator at 80 degrees Celsius. Then we get powder form of zinc oxide nanoparticle. In characterization, use a UV, SEM, scanning electron microscopy, ADX, and FTIR. And preparation of antibiotic base coated with zinc oxide nanoparticles. So about 20 milligram of zinc oxide nanoparticle powder dissolved in 1 ml of distilled water. So about 5 milliliter of suspension picked with the help of pipette and each antibiotic is well coated with it. So in the bitter bed containing coated base covered with the lid and kept in oven at 80 degrees centigrade. So the last one is this diffusion acid for zinc oxide nanoparticle coated and non-coated antibiotics. So the both zinc oxide coated and non-coated antibiotic disc were applied against each bacterium in Miller had the agar plates, and these plates are placed in incubator at 37 degrees centigrade for 24 hours. Then observe zone of inhibition. So the next one is a result. We discuss antibiotic sensitivity test. So antibiotic sensitivity test of all which isolate were tested against different antibiotics like ceftazidime, clarithromycin, cephradime, metronidazole, and amoxicillin. So in this, the results show that bacterial strain like Shigella, Enterobacter, Pseudomonas, Proteus, E. coli, Klebsiella were completely resistant against amoxicillin, metronidazole, and cephradime. In the next slide, Shigella, Enterobacter, and Staphylococcus are more susceptible as compared to other bacterial strains against equus and methanolic extract. The next one is characterization. So UV spectrophotometer analysis of zinc oxide nanoparticle. So in UV, they showed sharp peak that confirmed the synthesis of zinc oxide nanoparticle and also their involvement with the natural reducing agent present in the root extract. The next one is SEM, scanning electron microscopy. In this image of zinc oxide nanoparticle, observe loose morphologically aggregates, irregular and cubical with various size. The next one is FTIR. So FTIR analysis determines the presence of biological molecule, specifically functional groups present in the plant extract of zinc fission. So this analysis clearly determined that the link of plant metabolite to the synthesis of zinc nanoparticle, that the positively charged zinc ions and negatively charged biomolecules are responsible for the bond formation between these groups and zinc ion. Okay, the next one is bacterial activity of zinc oxide nanoparticles. So the data of antibacterial assay exhibit that most of the selected bacterial strain were highly susceptible to zinc oxide nanoparticles. So the bacterial strain showed different inhibition zone against very concentration of zinc oxide nanoparticles. So Klebsiella. And this Klebsiella was found to be most susceptible strain among all the strains. So it is showing highest zone of inhibition, 12 millimeter against 10 milligram of zinc nanoparticle. 5 milligram they show 10 millimeter of zone of inhibition, 4 milligram they show 9.8 millimeter zone of inhibition, and 2 milligram they show 2.8 millimeter zone of inhibition. The last one is conclusion. From the above study, it concluded that zinc official legume act as a potential reducing as well as capping agent for the green synthesis of zinc oxide nanoparticle. So this study will help to improve the quality of existing commercially available antibiotic against MDR isolates, material isolates. So zinc oxide nanoparticle showed magnifical antibacterial activity. Those biosynthesizing nanoparticle may be present in the prevention of many diseases. Okay, the last one is future recommendation. So new studies should be done on increasing yield of green synthesized nanoparticle, which is a major concern. So in future, more attention should be given to the biomedical application of green synthesis of nanoparticle. So however, further extensive research is required for biogenic zinc oxide nanoparticle for their exploitation in the field of nanomedicine. And thanks to all of you for attention. Thank you so much.